Today, we will be looking into confocal microscopy. How it works is the illumination from a laser through the objective is used to excite the fluorescent markers on the sample. Any light out of the focus planes is canceled out by the confocal aperture. Lights that do pass through the aperture will contribute to the image formation by the photomultiplier tube.
Next, we will take a look at the instrument. When using a confocal microscope, there are two phases, the extraction phase and the emission phase. During the extraction phase, a laser reflects off a beam splitter, and then that laser travels through an objective magnification lens where it reaches the specimen. Once the light reaches the specimen, we change to phase 2, which is emission. When the light reaches the specimen, it reflects off the specimen and then travels back up to a pinhole lens. The light that passes through the pinhole lens puts the light into focus and travels through the pinhole, where it later reaches the detector. The pinhole lens and the pinhole work in sync in order to only capture and focus light. Additionally, in the first phase extraction, there may be two mirrors that reflect the laser light in order to scan different parts of the specimen in order to get a whole picture.
So for the data, as shown in the figure, starting in the top left, the sample is scanned one section at a time. In one slice, the laser scans the sample in the X and Y directions, and each slice is reconstructed using software to create one image with all the slices, which is shown on the top right, with all the slices together. 3D imaging can also be available with optical sectioning, which scans the samples into slices from top to bottom, creating what's called a Z-stack, which can be shown on the bottom right, with all the slices top to bottom. Multicolor imaging is also available by using fluorescent light in specific dyes. Some samples and applications. Confocal microscopy is very good at imaging cells and tissues using depth, illumination, and color, which gives very good spatial information in the samples. On the bottom left, there's adipose tissue, which you can see all the light and the 3D. In the middle, there's 3D view of pollen samples, and on the right, there's dendritic cells.
Next, we are going to take a look at the resolutions that we can collect with a confocal microscope. When using a confocal microscope, we can collect high-resolution 3D images that emit out-of-focus light while also collecting the finely focused light. If we take a look at the left, we can compare a wide-field microscope with a confocal microscope. In order to understand this comparison, we can take a look at the bottom of the screen. A wide-field microscope essentially collects the same data, but it does not collect the finely focused light, it rather collects all of the light. Taking a look at the bottom, we can see, compared to the confocal microscope, where the light travels through the pinhole, that the wide-field microscope just collects all of the light, which means it collects the out-of-focus light as well, unlike the confocal microscope, which only collects the in-focus light. Looking above that, we can compare the two. Looking at the wide-field microscope, we can see that the image isn't very clear. You can see it's a little foggy and the lights aren't, or not the lights, excuse me, sorry. The colors aren't as defined. If we take a look to the right, the confocal microscope, on the other hand, is very clear. You can tell what's going on, you can distinguish the different colors, and you can also see what's going on in the background.